Okay, this is the second half of Chapter 7, where we were talking about this idea of second messengers, right? Remember the first messenger is the signaling molecule, the ligand that's going to bind to the receptor, typically at the cell surface. But then most of these pathways also involve molecules that are called second messengers, which are by definition, they're always small non-protein molecules that are helping amplify and carry that signal through all the parts of the cell. And so there's four common second messengers that I want you to know. There's what's called CAMP, which stands for cyclic AMP, DAG, which stands for diacylglycerol, and really as long as you can remember these abbreviations, that'll do the trick. IP3, inositol trisphosphate, and then just calcium ions. So I'm going to go through these briefly and give you a little bit more information here. So cyclic AMP, CAMP, is produced by this enzyme called adenylyl cyclase, and adenylyl cyclase will actually grab ATP, right, that energy-rich molecule, and alter its structure. It'll remove two phosphates from ATP and then bind the third remaining phosphate here, kind of back to the sugar. So you get this what's now called cyclic adenosine monophosphate, or CAMP. And I'll show you an example of this in a minute, but this adenylyl cyclase and hence production of CAMP is very often a part of these G protein linked receptor type pathways. Now, what does CAMP do? Well, it's got two main targets inside of cells. One is it will commonly bind to ion channels and open those gated ion channels. And then it can often also bind to protein kinases, so those enzymes, right, that like to add phosphate groups to other molecules, and basically activate those kinases. So it can start some of those kinase cascades that we were talking about, or phosphorylation cascades. The next two second messengers on that list are kind of tied together. So this is DAG and IP3, and they're linked together because both of those second messengers are actually produced from the same precursor molecule, which is this membrane-bound PIP2, it's called. And I'm just going to show you the image here. So I'll focus you in on the middle section here. So here's PIP2, and you can see it's this membrane-bound protein. So there's a portion of the PIP2 molecule that's kind of hanging off in the cytoplasm, and then there's a portion that's actually embedded in the membrane. And so both of the second messengers we're talking about here, DAG, which is up here, and IP3 down here, are basically just produced by splitting PIP2 in half. And PIP2 is split by this enzyme called phospholipase C. So if phospholipase C becomes activated, it will split PIP2 into these two halves. The half that's now cut loose into the cytoplasm becomes IP3, and the half that's still embedded in the membrane becomes DAG. And both of those are now considered second messenger molecules. DAG is going to be able to diffuse around within the membrane and potentially activate other membrane-bound molecules. IP3, since it's in the cytoplasm, can diffuse away. In this example, you see IP3 binding to a calcium channel that's on the surface of the endoplasmic reticulum, opens that calcium channel and allows calcium to flow out. And that kind of segues into the last second messenger, which was calcium ions. And this is really typical in cells. Usually the calcium concentration in the cytoplasm is very low. Usually cells will either pump the calcium into the ER or even pump it out of the cell completely. So as soon as any of these type channels open and calcium appears in the cytoplasm, that's kind of an unusual occurrence, and the calcium can bind to and activate other molecules in the cytoplasm and cause changes in the cell that way. So I think I just said all of this, but I did want to illustrate it may seem kind of boring. Okay, yeah, calcium concentration's low, yada yada yada. Well, actually, all of our lives actually began with a little calcium influx into a single cell. So here you have images. This is calcium imaging of a human egg, where blue color indicates low calcium levels and red indicates high calcium levels. And you can see in an unfertilized egg, so this is kind of a time series here. If we start in the upper left corner, we have an unfertilized egg, and you can see calcium levels are very low, right? That egg has been pumping calcium actively out of the cytoplasm. Well, upon fertilization, you see where the sperm binds up here in the upper right corner of this egg. The first thing that happens almost immediately is calcium starts flowing into that egg cell, right? So this interaction of sperm and egg has opened some calcium channels on the plasma membrane. We get this influx of calcium, and now that's going to basically set us off to the races in terms of embryonic development, right? That's step one. Calcium comes in, and before you know it, you're sitting on your computer having to listen to POBI lectures, right? Made out of trillions of cells and all these complex tissues and organs. So I realize, some of this may seem a little tedious or a little too detailed, but it is really, really important in terms of our biology and how we function and have developed as organisms. So hopefully you've got under your belts now some of these basics of these signal transduction pathways, right? Ligand binding to receptor, this idea of these cascades, the idea of second messengers and what they might be able to do. So now I want to talk just a little bit here at the end about what kind of responses can happen inside of cells and how cells could try to uh, exercise a little bit of control over these pathways. So let's start with this idea of cells being able to kind of tweak or control these signaling pathways. And there's really two main ways cells can do that. One is they could either increase or decrease some of the components of those pathways, those proteins or enzymes or whatever type of molecules they happen to be. Let's go back to the idea of one of those kinase cascades. If we get rid of one of those kinases, basically break down the enzyme that's part of one of those pathways, then that's obviously going to slow the whole pathway down or even shut it off completely. Or on the flip side of that coin, we could synthesize more of a particular enzyme that's in one of these pathways. Or we could do it a little bit more subtly, and instead of synthesis or breakdown, we just activate or inhibit them slightly. So it would be a little bit more of a measured way of adjusting these pathways. But hopefully you've gotten the idea when we were looking at some of these examples that a lot of these molecules that are part of these pathways can be turned on or off pretty easily. So, we have adding or removing phosphate groups is a very common way to activate or inactivate various molecules, including enzymes, right? So here we have an inactive enzyme, and we take a phosphate group from ATP via a protein kinase, and we add it to that enzyme, and now it changes its shape and its chemistry, and it becomes biologically active. And then we remove that phosphate group and we flip the switch back off. We saw something similar when we were talking about the G proteins, right? The G protein was inactive when it was bound to GDP, but we could swap the GDP out for GTP, and basically this is just adding another phosphate again. It's almost quite similar to example A up here, but we add another phosphate to GTP diphosphate to give us triphosphate, and we turn the switch on, right, remove it and switch it back off. And even with those second messengers, we could produce the second messengers, like cyclic AMP was made by that enzyme, adenylyl cyclase. But then there's another enzyme that I had mentioned, phosphodiesterase, that can break down cyclic AMP and switch it off or remove it completely. So lots of these on-off switches. And these are all just giving the cell a little bit more control over some of these pathways. Okay, so let's talk about end results of these signals. One that I had already mentioned is changing gene transcription, right? So transcription is the process of making an RNA copy of specific genes, and then that RNA copy would be used to make specific proteins coded for by those genes. So essentially, when we say we're changing gene transcription, really what we're doing there is altering what proteins the cell is making. So that's a very common way of causing something new to happen within a cell, right? As we make a new enzyme, make a new protein, or maybe stop making one that was already being produced. But a couple of other common effects could be opening ion channels, or simply altering the activity of an already existing enzyme. And so for any given signaling pathway, right, one or more of these might happen just depending on that particular pathway. Just to give you one example here, so this is basically how your sense of smell works. So you would have some odorant molecule, right? Hopefully it's a good one, not a bad one. Comes in, and these odorant molecules will bind to G protein linked receptors on the surface of cells in your nasal passage in your nose. The G protein linked receptor gets activated, right? Activates the G protein. G protein will activate adenylyl cyclase, which produces the second messenger, CAMP, and then CAMP that's produced will bind to this CAMP-gated ion channel. That channel will open and it'll allow calcium and sodium ions that were concentrated outside the cell now to flow in, and that the presence of those ions inside the cell now is going to stimulate nerves to send signals to your brain that says, hey, I smell something good or something bad, right, depending on the particular odorant molecule. So the end result in this case would be nerve signaling gets initiated. I already mentioned this idea of altering enzyme activities, but just keep in mind, enzymes are proteins that are catalyzing chemical reactions, and their activity can be either enhanced or inhibited by tweaking the shape of the particular enzyme. So obviously if we enhance an enzyme shape, enhance its activity, whatever reaction it's catalyzing is going to happen more frequently. So that's a common way of altering the biochemistry, the biology of a cell. And then I've already mentioned this idea of altering transcription, so I won't dwell on that, but hopefully you've got that idea. Okay, last thing I wanted to mention here is there are ways, and I sort of already touched on this in previous chapter, but there are ways that cells can communicate directly without having to have these signals that are coming from outside of a cell. So this is direct cell-to-cell, cytoplasm to cytoplasm communication. And this can happen in animals through gap junctions, which I mentioned in chapter 6, or in plants through these structures called plasmodesmata, which I think I may have mentioned briefly in chapter 5. But regardless, just to remind you, right, gap junctions are one of these types of cell-to-cell junctions, and they are considered communicating junctions because the gap junctions literally create a little pore through the plasma membrane that connects the cytoplasm of two adjacent cells. So you could have some small signaling molecules, maybe some of those second messengers, for example, right, calcium ions or cyclic AMP, what have you, that could now directly flow from the cytoplasm of cell 1 into cell 2. So we're able to pass a signal very quickly through cells connected by these types of junctions. Yeah, so one example here, you have lots of gap junctions in the lens cells in our eyes, um, and really that's a good way to be able to send signals very quickly, cell to cell, without having to have bloodstream nearby, right? We don't want a lot of blood vessels in the lens of our eye, or it's going to obscure our vision. So sort of an alternative way to pass signals. And then in plants, we don't have gap junctions, but we do have something sort of similar called plasmodesmata, um, which are really channels connecting the cytoplasm of one plant cell to another. In this case, the plasmodesmata are a little bit longer than the gap junctions because they have to go all the way through the cell wall that would be between two plant cells. And they also look a little bit different because there's almost always endoplasmic reticulum that goes through plasmodesmata, so it's kind of unusual. You have the ER in one cell, like physically connected to the ER in its neighboring cell. So you could have molecules actually inside the ER moving through from one cell to the next, or there is a little bit of space around the sort of ER tube that goes through the plasmodesmata, so there's some space around that which small molecules could pass through as well. But again, you could have signaling molecules moving from one cell to another. So anyway, that is it on cell signaling. Hopefully you have feel comfortable with some basic understanding of how these signaling pathways work. And in the next section, basically chapters 8, 9, and 10, we're going to begin talking about some energy concepts, and that's going to lead us into cellular respiration and photosynthesis.